Assalamu. We are covering part 6 of testing universal common ancestry, and with me is the philosopher Paul Nelson. Welcome, Paul.
Thanks for having me. I always like being here.
Yeah, it's a good series we have going on, and I've been advertising this as the most thorough universal common ancestry course on the internet, and I don't think that's an overestimation.
You know what? I think you're right. Now that we have 6 parts and with another one to come next week, it really is like a mini course in evaluating the logic of universal common descent.
Yep, that's right. And I've made a playlist on my channel, so anybody can go there and check it out. Also, one very important thing is if you are tuning in now and you're not understanding what we're going to be covering, don't worry. Some of the background knowledge is from the previous live streams, but it's still worth you watching and learning what you can actually learn from today's live stream. So, Paul, over to you.
Great. I should indicate that I will be providing the citations, a complete list of the references to Subor after this taping occurs, and he will put those beneath the video on YouTube.
Perfect.
So, in previous sessions, I've said that certain formulations of universal common descent are untestable, and I'll talk more about that today. But someone with knowledge of the evolutionary biology community might reply, well, Paul, I know that some prominent evolutionary biologists, such as Carl Woese or W. Ford Doolittle, have rejected universal common descent. And the question is, of course, why, or more precisely, how? If the theory is untestable, as you, Paul, have been arguing, then how could any biologist like Woese or Doolittle have rejected the theory via testing? So that will be the question we'll explore today. And we will use the ribosome as our sort of grand example at the end, but we'll get there via some other examples first. So we need a model, a philosophical model of testing, and I'm going to use the classic model of modus tollens, which is that theories entail or make predictions empirically that have empirical content, and then we can go out and check them, and we check to see if those observations, namely the predictions, obtain. If they do, the theory is provisionally confirmed. So this is philosophy of science 101, right? And I would, in fact, I'm not going to do so today, but I would argue that almost any model or rational reconstruction of testing will boil down to some form of modus tollens. But again, I don't want to go into that today. Of course, then the test will be the observations, the entailed predictions do not obtain, and then the theory is potentially disconfirmed or falsified. So a very basic but quite useful model of theory testing in science. And this is something to which we'll hark back throughout the presentation today. Now, universal common descent, so biological theory, cannot be tested directly. And the reason for that is the theory doesn't rule out anything. And this has been known by biologists who look at the question for some time. In fact, I pulled here just one example of this from the work of Lars Vogt, who, when he published this paper, was at Harvard. He's now in Germany. But in this paper in the journal Cladistics in 2007, Vogt pointed out that a given tree hypothesis, and of course that would include universal common descent as sort of the ultimate tree hypothesis, is logically congruent with any specific observable evidence of character state distribution. In other words, a given tree in combination with descent with modification as background knowledge does not prohibit any specific character state distribution pattern. In simpler language, if you have a tree of descent where you have some universal common ancestor, or perhaps you're looking at just part of the tree, so let's say common ancestry for the eukaryotes or common ancestry for the animals, for the metazoa, Vogt's argument is you could have any imaginable distribution of characters, that is, features of organisms, along the tips of that tree, the termini of the branches, and common descent as a hypothesis would say, well, that could happen, right? It may be less probable than some other tree, but any imaginable distribution of characters, given the nature of evolutionary theory, could occur. So Vogt goes on, there's no deductive link between any tree hypothesis and any specific character state distribution. One cannot think of any observation which, in case it would represent a true statement, would allow one to conclude the falsity of a clade or a given cladogram through modus tollens. So again, you've got a tree hypothesis of relationships among some group of organisms. The tree itself does not deductively entail any particular distribution of characters along the tips. And remember, it's the tips that we observe. So he argues, you can see it right there in the title of the paper, cladograms, and we can generalize that to evolutionary trees, are unfalsifiable. So how do we test it? Well, we test universal common descent with the help of the rest of biology. The standard formulation of universal common descent is very brief. You can do it in a single sentence. All living things on Earth descended from Luca, the last universal common ancestor. Now, that formulation is very attractive because it's so general. In fact, in the case of biology on planet Earth, it is one of the few genuinely universal statements that biology makes, because it applies to every living thing, whether extant today or alive in the past, the 4.5 billion year history of the planet. So its universality makes it enormously attractive to scientists as a proposition, as a theoretical claim, but it doesn't rule anything out. Of course, any theory that rules out no observations cannot be tested by observations. And again, this is very basic philosophy of science, and I am not going to go into the epistemology here today. But if your theory is consistent with everything that you see or can imagine seeing, it's not capable of being tested. So that's a problem. What do we do in a case like this? Thinking now as evolutionary biologists, we couple universal common descent to helper or auxiliary theories drawn from the rest of biology to enable specific predictions to be made. And then we can compare those to observation. So you see here in the cartoon, we have universal common descent coupled to some auxiliary theory drawn from the rest of our biological knowledge, which enables us to make testable predictions. So what does universal common descent supply? Well, it supplies a geometry, a formal geometry. Luca must have existed here at the root of the tree of life. Luca is not or cannot be identified with the origin of life. There may have been, in evolutionary theory, some significant passage of time between the first origin of life and Luca, but it is the unifying organism that is a member of the set of ancestors of any organism on Earth. And moreover, and this is the key logical point, it's the only member present in all such sets of ancestors, if you do this with some basic set theory. So there's our domain of universal common descent, springing from Luca. Now we can couple that to everything else we know in biology. Okay, and that then lets us make predictions. And I'll give you a few examples before we turn to looking at the structure of the ribosome. So this, if you go into the literature, both the primary research literature, but also textbooks and review articles, discussions of the empirical support, the empirical content of universal common descent, you will see this instantiated, where universal common descent is coupled to some other part of our biological knowledge, and then that gives us predictions. And typically, the logical form of those predictions does the following. You take a rule or a prescriptive generalization derived from what I'm calling AUX, right? The auxiliary theory. And because universal common descent gives you a domain, you apply that rule across the entire domain, or the geometry of relatedness. So that's what the linkage does. It takes some rule or prescriptive generalization, X will not happen, and it extends it across the whole domain, both for things you've observed. After all, that's where you got the rule, right? From looking at cell biology or looking at developmental biology, if we talk now just about the animals, and you apply the rule across the whole domain. So here are some examples of such rules. The first one now, genetic code cannot vary. That was something I learned as a student, which is now no longer held to be valid, right? That rule has gone away. But when I was a student of biology in the early 1980s as an undergraduate, and then a graduate student from the mid 80s to the mid 90s, was generally thought to be the case. Not so in 2022. This second rule is still the case. All cells, all free living cells require ribosomes. Viruses are defined in part by not having a ribosome. They have to hijack one from a free living cell to turn their genetic information into proteins. And this third rule is also still held to be the case, that to have a proton gradient, which was necessary for metabolic activity and really to maintain the homeostatic capabilities of any living thing, from Homo sapiens all the way down to E. coli, boundaries are necessary. A cell wall or a membrane is essential to maintain a proton gradient so that you can do productive work as an organism. Okay, so we take these rules and we, barring the first one now, which is thought no longer to be the case, we couple them to universal common descent, and then we can make claims that extend both, can extend across a domain where we actually haven't yet observed lots of things. Now, these prescriptive generalizations, these rules, bring strong theoretical constraints along, right? Because they tell us what is possible or impossible in any hypothesized pathway of evolutionary transformation. And this logical consequence has come to be known as the principle of continuity, which when you see it in action, you will see it really everywhere in evolutionary theory. So it's simplicity itself, beautifully simple but powerful principle. If we hypothesize a pathway of evolutionary transformation from A to B, so we say the features of B, the characteristics of B, organism B, derived from starting point A along the pathway there, then every point in that pathway must be biologically possible. Now, that may seem to be a truism. In fact, it's very powerful because our knowledge of the blue arrow, if you will, turquoise color, I guess, from A to B, that is independent. Our knowledge of whether that pathway is possible is logically and evidentially independent of our hypothesis of transformation, the white arrow, A to B. And it's that independence condition that makes the principle of continuity powerful, that gives it actually probative strength when we come to evaluate evolutionary hypotheses, as I'll show you in just a moment. So here's Leslie Orgel. In 1968, he wrote a widely cited paper on the origin of the information transfer system familiar to us from terrestrial biology, the DNA, RNA, protein system that every student learns in their genetics or cell biology course. And he's in this paper, I think it was published in the same issue of the Journal of Molecular Biology, if I'm not mistaken, the same issue where Crick, Francis Crick, gave his frozen accident account of the origin of the genetic code. I believe they were, those papers were in the same issue. I'm not 100% sure about that, so please check that before you tell anybody else. Anyway, Orgel says in that paper, I shall be guided by a principle of continuity, which requires that each stage in evolution develops continuously from the previous one. Okay, which is just a restatement of what I showed you on the slide just a moment ago about those two pathways. He goes on. It is very difficult to see how a totally different biological organization could have undergone a continuous transition to the nucleic acid system with which we are familiar. Well, what does he mean by that? Well, here's what he means. At the time, in 1968, and this is also the case today, there were hypotheses out there in the evolution literature that the familiar system B, based on DNA, RNA being transcribed, information transcribed from DNA into RNA, RNA then translated into protein. The system we all know, the features of that system were not originally present when life got up and running. So that's A here in my cartoon. There was some other system not based on nucleic acid that was the first system capable of Darwinian evolution. And at some point along that pathway, all the information accumulated here, specifying cell function and so forth, was transferred over to the DNA, RNA, protein system that we know. And Orgel looks at this and he says, that can't happen, right? And that claim of that can't happen, I'm calling here on the slide a gulf of biological impossibility, is based on what Orgel knows about information transfer in cells today. So he's got a body of knowledge about the functional limits, the functional constraints on information transfer in extant cells. And he sort of retrodictively back predicts that system, whatever its antecedents were, must have had continuity all along this pathway. And he says, if we had to have a completely different chemistry, I don't see how this could happen, given what we know about how this system B actually works. So he's taking these rules and saying, I'm sorry, I can't buy this particular A to B hypothesis if it requires me to transfer information from one kind of chemistry to another, maintaining viability all along the way. So here you see the principle of continuity in action as a regulative principle, ruling out an evolutionary hypothesis. Okay. Now, key point that will carry into the rest of the presentation. When you, as an investigator, are met with anomalous observations, you have the option to retain your auxiliary theory, to privilege that over universal common descent. Okay, and this is, I'll give you some examples in a moment. This happens, I think, for people who are not in evolutionary theory on a professional basis, they're surprised when they learn how often this actually happens when an investigator says, I actually know more about this, my auxiliary theory, which gives then the body of knowledge that I use to ground my auxiliary theory also gives me a principle of continuity that I then take over to universal common descent, and I say the geometry that this requires cannot be the case in light of what I know here. Okay, and this is unrelated to the intelligent design debate. All right, this is actually everything we're talking about so far is still entirely within evolutionary theory on a naturalistic foundation. The investigator will say, I actually know a lot more about this, and the geometry that this entails, the universal common descent entails, cannot be the case. And they jettison universal common descent. So let me give you some examples of this as it plays out in the recent evolutionary literature. And this is just a selection from a very much larger pool of cases. And for the sake of time, I'll just give a few. But I want to show you how this works in action. So Massimo De Giulio, I hope I'm pronouncing his name correctly, is an evolutionary theorist in Italy who works on the base of the tree of life. And he has a long series of papers in the Journal of Theoretical Biology dealing with how the major domains of life, bacteria, archaea, eukarya, came to be. And in 2018, he argued that when these cellular domains arose, they did so independently of each other. They did not have an organismal, what he calls a genote. They did not have a genote, namely an organism with DNA, RNA, protein that we would recognize as their common ancestor. And he says the reason is fundamental cellular types, such as the architectures of these different domains, archaea, bacteria, eukarya, cannot be transformed into each other. They are aboriginally distinct. So he says that if your hypothesis, your evolutionary hypothesis requires the transformation of one fundamental cellular state into another, he says, this seems impossible to accomplish. So you see there, in light of the theoretical schema I gave you on earlier slides about the principle of continuity, you see how continuity here is, again, performing this regulative role, ruling out hypotheses on the basis of their apparent impossibility. He goes on, there are strong arguments that these fundamental traits that define the domains would have to be changed in a way that can't happen. This transition among domains of life cannot have been realized. And you can actually see this, as I've said in previous presentations, every figure in a paper is a summary, a visual summary of an argument. You can see this in his figure here. A progenote is not a cell that we would recognize. One of the things that troubles me about his work, De Giulio's work, is to my satisfaction, he never really adequately describes what a progenote would be, right? And it's a problem, in fact, for origin of life research in general, that as we leave the cellular, the world of cells as we know it and begin to try to describe cells as they must have been, they lose the features that actually define them. And they become very ephemeral. It's hard to actually say what a progenote is. But the important point of this figure is these are not organisms that we would recognize. And you'll notice that the genotes, cells that we would recognize, these are arising independently of each other from this hard to define, rather nebulous state of ancestry. So these are crossing this threshold into what Massimo De Giulio calls the genotic era. They're not in the figure. He's not tracing them to a common ancestor. This transition into the living state that we would recognize as cellular having the features of cells that we know is happening multiple times independently. So he says, the argument that there had to be an organismal common ancestor, he says, is an assumption. And as such, it might be false, above all for the deeper nodes in the tree of life. So again, you can see continuity playing a role here, breaking up Luca into something that Giulio actually doesn't describe, but he says when cells as we know them come to be, they do so multiple times independently. Didier Raoult is a microbiologist, very productive microbiologist in France, who ran into some notoriety during the COVID pandemic for promoting hydroxychloroquine, I think it is. I'm sorry, I don't remember exactly the name of the drug, as a therapy for COVID. But we can set aside that he's a very thoughtful, productive microbiologist. So set aside the COVID notoriety. In 2012, he and I believe a postdoc published a paper where they said the tree of life, that's what TOL stands for here, the tree of life concept is contrary to our current knowledge. And his argument for that is that the number of unique genes present in every bacterial genome, so-called orphan or taxonomically restricted genes, is so high, 10 to 15%, he says, of every species, bacterial species that's examined has this very high fraction of unique genes that, he says, cannot be reconciled with a single tree picture. So in the same paper, he and his co-author have this very striking figure here where they show what they call the rhizome. A rhizome is a biological structure with that lacks a single root, has tangled interconnections. And these are bacterial groups here at the bottom, bacterial genomes, where the content of the genome is drawn from all kinds of different groups in this kind of crazy pattern here of a network of borrowings. And you can see, again, my aphorism, a figure is a summary of an argument, a visual summary of an argument. The argument here is there is no tree, right? You've got just a wild sort of Roman orgy of horizontal transfer going on, and it's impossible to derive a single tree from the data. Another example, in many ways, I think the most striking example is the work of Eric Baptiste from Paris. And again, I'll provide the citation so you can look up this talk. These are slides from a talk that he gave. Baptiste is a leading critic of the single tree of life geometry within evolutionary theory. And he has multiple lines of argument, complaints about practices and the handling of data within standard evolutionary theory that he said bias investigators without them even really being conscious of what they're doing towards the single tree geometry. So for instance, here on the left, we have the total data set, genomic data set for some group. And what Baptiste is troubled by is the practice of eliminating data from that data set, excluding genes that have a patchy taxonomical distribution, right? So those are dumped out of your analysis. Or genes that have more than one copy per species, that have paralogs. And paralogs are problematic for phylogenetic reconstruction, so they get dumped out. Or genes, genes whose the trees that would follow from using those genes in a phylogeny reconstruction would give you relationships that are, as he puts it, obviously odd. You already know they're wrong, right? You know that can't be the correct phylogeny. So if that gene is telling you, giving you an odd phylogeny, out it goes, because it's giving you a misleading signal. And you get this filtering, what he calls massive filtering, where data are dumped out, eliminated from the analysis to give you, right, the maximally consistent signal. And Baptiste says, what biological sense does this make? What's happening is the preconception of universal common descent is filtering data, and the data are not being allowed to speak for themselves. He says he finds this highly objectionable. And he said the fact is the incongruities presented to biology in the genomic era, so roughly from the mid-90s until today, we're looking at about a 25-year window, very surprising period when a lot of data that were unexpected in the 20th century, the second half of the 20th century, where phylogenies were constructed on the basis of single proteins, single genes and proteins. And now, of course, we have whole genomes and tens of thousands of them. In that period, Baptiste said, increasingly evolutionary biologists began to question the single tree picture. So he was a postdoc with W4 Doolittle in Canada. And this is Doolittle's drawing of his understanding of the evolutionary history, where you notice there's not a single root here. This is multiple trees arising from an ill-defined prebiotic milieu. And what happens in a case like this, Baptiste says, is the question changes. The question being put to nature changes from how does the tree of life look to is there a tree of life? So what he calls a fundamental shift regarding this geometry. When you ask this question, you presuppose that the tree of life is there to be discovered. And Baptiste says what's happened in the light of these anomalous data coming from whole genome sequencing is the question now has shifted, is there a tree of life? Now, this, I think it's a really interesting talk. When you see it, you'll understand what I mean. But I think this slide for me was one of the most interesting from Baptiste's talk, because what he points out is that if you're looking at species 1, 2, and 3, and you're asking what was their common ancestor, and you're looking at different molecular markers. So this would be a eukaryote. If it had a nucleus, it would have nuclear pore proteins, small subunit ribosomal RNA, polymerases, and so forth. These molecular markers are not giving you the same histories, okay? And the problem is they are pointing to common ancestors, molecular common ancestors at different points in time, meaning there was not a single organism that possessed all of these molecular features that was capable of giving rise to all of them congruently. So he calls this fictional last common ancestors. And he bases this on this point on the work of Jeff Lawrence at Pitt, and I believe one of his doctoral students, Adam Retchless. So they had noticed this pattern. Sorry, I just want to go back. They had noticed this pattern in bacterial groups that they were looking at. And they wrote a paper where they had one section, a whole section of the paper, they say, there's going to be an inevitable failure of the tree of life. And they say, these ancestral populations simply did not exist. If we force the data, to tell us this, the data refuse to cooperate, right? there was not a population within the tree of life that could have given rise to what they observed within different bacterial groups, what they call fragmented speciation. And they said this kind of reticulation in relationships effectively invalidates hierarchical classification. Well, if the tree of life is anything, it's a hierarchical classification. So again, unrelated to the intelligent design debate, really, I guess you could say orthogonal to that, is this, say somewhat awkwardly, this coming apart of the tree of life. Now, I got to move on to ribosomes, but what I want to show you is these examples and others that are too numerous to mention show that when a biologist is confronted by this, universal common descent plus auxiliary theory failed prediction, many evolutionary biologists now will make universal common descent pay for the observational failure. All right, we come to the ribosome. And, really, to my mind, the most interesting molecular machine out there. Far more important to biology than some of the, poster child examples of intelligent design, such as the bacterial flagellum. We have a figure here showing on the left the small subunit, on the right the three-dimensional form of the large subunit. And then these are secondary structure representations of the very large RNAs that are that provide the skeleton and the functional site, the peptidyl-transferase center in the large subunit. So this is going to do the decoding work. The small subunit is going to do the decoding work, and the large subunit will do the assembly, taking the information that's on the messenger RNA strand that's being decoded by the small subunit, and in the large subunit, the transfer RNAs carrying amino acids will form peptide bonds and beautiful, elegant story and make proteins and so forth. Okay, now, a figure like this focuses on similarity, understandably, right? Because there are a lot of similarities here. When you just visually just look at this huge RNA, right? You can see that if we could superimpose these, there would be many regions of homology between them. Of course, there are regions that are not homologous, illustrated here by these black portions of the diagram. Here, when we get to a eukaryotic ribosomal RNA for the large subunit, you've got some pretty dramatic extensions here that are not present in these two prokaryotic forms, bacterial and archaeal respectively. But the figure itself draws you towards a view of conservation, right? Evolutionary conservation of form and function. Now, as I've said in previous talks, there's a reason for that. There is a deeply held preconception about the pathways, evolutionary pathways leading to the formation of large, complicated molecules like this very large RNA, that says it could only have happened once. So I've used this slide, this example in previous talks. The reason I'm coming back to it is it does such a beautiful job of showing the point that I'm trying to make. So Jonathan Eisen is a professor at UC Davis who teaches evolutionary theory, among other things. And in one of his lectures to undergraduates, he's explaining to them the difference between homology, that similarity that gives you reliable history, right? That you can use to construct evolutionary trees because the structures you're looking at shared a common template, a common material template at some point in the past. So homologies are what you want to build reliable evolutionary trees of relationship. Homoplasy, by contrast, is misleading similarity. There's similarity, all right, but the similarity is not giving you reliable history. So he's trying to get the students to see there's an important difference here, and you want to look for those features, those characters and systems that are going to give you homology. How will you know, however, that it's homology? Well, he uses the ribosome as his example. And if you go and download the whole lecture, he compares the ribosome to other molecular features, which he says you might get those multiple times independently. Just to be an organism on planet Earth, you may need to have lipids. You may need to have nucleic acid, right? In which case, it would be a potentially homoplasious. There's an awkward adjective. It would be a potential homoplasy, not giving you reliable history. Why do we know, or how do we know the ribosome is giving us reliable history? Because there's no way it could have happened more than once. Which I'll have to tell you, if I'm thinking about this as an evolutionary theorist, I look at that very large RNA that gives you the skeleton and the active site of the large subunit. I think, yeah, there's no way you could get that more than once. Well, if that's true, if this pathway right here to the first ribosome is a very small probability Markov chain, right? Mathematically analogous to a draw from a lottery cylinder, where you've got maybe, I don't know how many, 2,000 balls, numbered balls, right? And you give it a spin and you take out the balls, align them, read off that sequence. You will never get that sequence again if it's a true lottery cylinder, right? So this is a very small probability pathway. We're just lucky to have it at all. And Eisen concludes the only possibility, therefore, is a single tree. The ribosome is going to give you that single tree geometry. Now, if you think about this carefully, you should realize that in order for this to be true, in order for this geometry to be true, all the segments in this pathway, from original ribosome to its descendant forms, the offspring, so to speak, the ribosomes that evolve along the branches of the tree of life, all the segments in this pathway must be biologically possible. Again, that may seem to be a truism, but that's what the principle of continuity tells you. No miracles. It's got to happen via evolutionary processes. Which raises the question, quite inevitably, what evidence do we have that these structural changes, right, the differences that we see here, the regions in black that are not shared, that are not homologous across the secondary structures here of this very large RNA, what evidence do we have that those occurred via undirected mutations to ribosomal RNA? Now, this is a place where a pattern claim, a pattern about homology in this case, intersects logically with a process claim like natural selection. Okay, so I'm going to go into just very briefly into the logic of natural selection so you can see what the key parts are of this process. And it's expressed as a conditional. So if within a species or population, the individuals vary in some trait, so we have to have the condition of variation, leave different numbers of offspring in consistent relation to the presence or absence of that trait, call that the condition of selection, and transmit the trait faithfully between parents and offspring, the condition of heredity, then natural selection will occur. The frequency of that trait, I'm calling it here trait Q, will differ predictably between the population of all parents and the population of all offspring. So this is a standard, it's empirically sound or conceptually sound. This is not circular. You'll notice that the term fitness has been eliminated from this formulation, which is a good thing. That's another lecture. In any case, this will be standard evolutionary theory, novel variation will arise via mutation. So the question we're asking about those differences in the ribosome is, if those arose as undirected mutations, and those mutations were fed into this process of natural selection, were the three conditions satisfied? If natural selection, just to pick one particular evolutionary process, was causally responsible for giving us the differences that we observe in ribosomes today, were these conditions satisfied? In particular, was this condition C, transmission, heredity, was that condition satisfied? So you can think about it more simply this way. These are the requirements of natural selection. You cannot invoke natural selection as a causal process if you can't provide evidence for all three of these. They are like three legs of a stool, and you need all three in order to employ natural selection as a causal process and evolutionary explanation. So this isn't restricted. This point that I'm making, by the way, is not restricted just to natural selection. Any evolutionary process, really any imaginable evolutionary process, has to fulfill these requirements. You have to have novel variations arising ultimately from mutation. Those variations have to make a difference of some kind, functionally or in some other way. And you've got to be able to pass them on stably. In other words, they can't be lost. There has to be a permanent, temporally asymmetric, where from past to the present, there's an irreversible change happening. You have to have variations. They have to make a difference, and you've got to pass them on for keeps. Well, this would be true for drift. This would be true for so-called constructive neutral evolution. Any evolutionary process you can imagine has to fulfill these requirements. All right. The question before us is, what is the evidence that this could have happened, that these differences in ribosomes could have arisen via undirected mutations? Now, I recommended this YouTube talk last time, and I'll provide the citation again so you can go watch it. Lauren Williams is a really thoughtful, creative origin of life and ribosome researcher in Georgia. And I think very highly of him because, first of all, he's funny, and he's a good teacher. So watching this YouTube video will definitely be worth your time. So he presents a nice lecture on the origins and evolution of the ribosome using his sort of accretion model, where the core of the ribosome accretes differences over evolutionary time. It's a very compelling model. But then he comes to a Q&A at the end. You can see where the here on the slide, it's the video is almost over. He's doing a Q&A with the audience, and someone asks him, can ribosomes be re-engineered in vivo? Right? So you've got, let's say, yeast, or you've got E. coli, and you want to tinker with their ribosomes in living cells, right? Not in a cell-free system, but actually, you've got a cell that's got to make a living for itself. It's got to stay alive. And I was very struck when I watched this video the first time by Williams' answer. And he said, you really can't do this, right? So what you see here on the slide is a quote, or a series of quotes from Williams, where he's saying it's really hard to do. It's hard to modify ribosomes in vivo because cells don't like it, right? This is the core of their biology, being able to turn their nucleic acid into proteins in a way that's viable, in a way that's functional. And he said it's really hard to tinker with that without killing the cell. He points out that in yeast, there's a whole chromosome that's nothing but ribosomal RNA sequences because of the importance of this machine to the living state. And he says, well, why can't you tinker with them? Well, because the centrality of the ribosome to everything else that's going on in the cell is so great that if you tinker with the ribosome, you crash the whole system. So I'm sitting there, watching this YouTube video, and I think, wait a minute. If undirected mutation was causally responsible for the differences that we see in extant ribosomes, why should it be so cotton picking hard to change them? Right? You ought to be able to subject them to mutagens or go in with some cell engineering system and tweak these sequences and have functional ribosomes come out the other end. So it struck me that as much as I respect Williams, and I really do, there was a rather large gap here in his understanding of the evolutionary outcome of some process that gave us these differences. If it's natural selection, you've got to use the logic of natural selection, starting with random variation. And I am in the midst, it's a long-term project of going through the literature of ribosome evolution, ribosome function, ribosome, how ribosomes fail. There are many mutations in ribosomes that have disease consequences. Don't have time to talk about that today. It's quite interesting, though. I'm in the process of plowing through the literature, going back decades, to try to find the evidence that undirected mutations to these structures are viable. Now, here's why this matters. And we're really now coming down to the last bit of the talk, but really it's the most interesting part. So I saved the best for the end. As I said earlier, when you see a diagram like this that stresses conservation, you think, well, that's got to have a common ancestor. I mean, I'm pretty much on any given day of the week, I'm thinking half the time as an evolutionary biologist. I look at that and I say, wow, that, look at the conservation of structure there. Yes, there's some differences among the domains, but there's a lot that's the same, okay? Once I got away from diagrams like this and from I've said it now for the third time today, the arguments that these diagrams instantiate, and got into the ribosome literature, I discovered that ribosomes are much more diverse than I realized. So here's a beautiful figure from a paper. Lauren Williams is the senior author. I think there are 5 authors on this paper. Really, just someone put a lot of work into, or more than one person put a lot of work into this figure. This is the secondary structure of the E. coli small subunit here on the left and the large subunit on the right. And very helpfully, the secondary structure has been color-coded with respect to what these different regions of this RNA are doing. Okay, so some of these regions are multiply colored, right? Right here, these regions have multiple colors because they're involved in more than one function. Now, there are proteins, lots of them, in fact, that associate with these ribosomes. When the ribosome is assembled, it has its RNA core and its RNA skeleton, but it's decorated with all kinds of proteins. And the proteins are not shown here. This is simply RNA, but they're there. Now, here's another figure from the same paper, and you can see there's a common ancestry geometry being presupposed here. Somewhere down here was LUCA. And then you have the ribosome branching into its eukaryotic, archaeal, and bacterial representations or forms. And this is a Venn diagram, and the region of overlap here in black, these are the universally shared proteins. When you come over here to the eukaryotes and the archaeans, you have proteins that are present only in eukarya here, you have proteins present in archaea. And very interesting, you'll notice that there are no only archaea, at least at the time of the publication of this paper. I'm not sure if that's still the case today, but in 2020, when this paper was published, there were no exclusively archaeal proteins. If there's an archaeal protein that's not in bacteria, it's also going to be in eukaryotes. So these are shared. Okay. Bacteria have a set of their unique proteins. Now, you look at a diagram like this, you think, well, these must be all homologous here because they're shared among the 3 domains. That is the expectation that I had when I saw this paper, that these are universally conserved. They're going to be homologous across 3 domains. All right. Let's look at 3 of them. UL15, US12. So L is large subunit. That's what the abbreviation here. U is universal, large subunit, protein 15. This is a small subunit, protein 12. And we'll look at 14. Now, this is only one thought experiment in this talk. I did a lot of them in previous talks in this series, but this is the only one. What would you expect about the structure of these proteins given this figure? Okay, so they're in this black region, they're shared, they're universal. What would you expect when you look at the topologies of those proteins? Couple universal common descent to what you know about how ribosomes are essential. You can't be a free living cell without a whole bunch of them. All right. And they're rather intolerant of tweaking, as far as we know. My expectation was conservation, absolute conservation. Well, that's not true. All right. So here we have along this side of the diagram the names of those proteins. Here's UL15. This is the large subunit, number 15, US12 and US14. And across this part, horizontally across the diagram, we see these proteins as they're found in different groups. And what you'll notice is they have lots of details that are not shared at all. All right, so there's a lot going on in this figure, but what you need to notice is that this gray conserved region is a minority of what's actually present when you look at different groups. So in mitochondria have their own ribosomes, and they're very weird compared to the ribosomes that you see in bacteria, archaea, and eukaryotes. So in each case, as you go across the figure, you'll see that there are regions that are not shared. Okay. Now, I did not expect that on the basis of this figure, that there would be that much diversity in these proteins. And when you look actually across the 33 of the so-called universally shared proteins, and I realize, again, this is a lot going on in this figure. I'll explain what's here. What you see is some things that are universally conserved. That's this region of light pink here. And then this region in red, these are the features that are found in a single domain. So we have Homo sapiens here, here's mouse, here's yeast, here's another kind of yeast, here's Drosophila, the fruit fly. So these are well understood model systems in eukarya, and we come across all the way here over to mitochondrial ribosomes. And as I said, mitochondrial ribosomes are weird, so you can see they have a lot of unique features that are not present elsewhere. What a figure like this, a beautiful figure like this, tells you is, yes, there's conservation. There's also a lot of uniqueness, and the uniqueness may be functionally critical to these ribosomes. So this paper gave me goosebumps, not an exaggeration. In fact, I can see the Starbucks where I was having coffee, that was probably the caffeine was hyping me up even more. When I read this paper, I got goosebumps. And for that reason, I tell you, go out and read it. Because in this lab at Yale, this group looked at these proteins and said, you know what, we are missing a lot of their diversity, which may be functionally critical species by species. So I'm not going to steal their thunder. I've already borrowed 2 of their figures. Please go look this paper up, and the references will be in the notes on the YouTube video, so you can do that. I'll stop here. I've gone on for about an hour. I apologize. I was hoping to be done sooner than that. But I want to leave you with 2 intuitions that I think really ground mainstream biological thinking about the ribosome. The first intuition comes from looking at a figure like this. And the complexity that is here, and the intuition that's triggered in me when I think as an evolutionary biologist is, this could not have happened more than once. This level of complexity could not have happened more than once, given some unknown natural pathway to these very large RNAs with their functional specificity. And that interfingers analytically, or at a deeper intuitive level, with this kind of geometry, right? Question is, if this could only happen once, how do I know it happened at all? Right? Singularities, mathematically, and in terms of their evidential support, a singularity, a single pathway, is hovering right on the edge of not happening at all. And that is a precarious position for a science of the past to be in. Okay, and I would say that if I were 100% an evolutionary theorist, not half-time, as I am most days, I would not want to live there. Counting on singularities, counting on one-off events where I'm just happy that it happened at all. But I think the reason that this geometry dominates evolutionary thinking is because this original pathway seems to be so unlikely. And I'll stop there and have a little chat with Subor. And thank you for being so attentive.
Thank you so much, Dr. Paul. I mean, the last slide just reminded me of that old saying, give us one miracle, we'll explain the rest. So you covered quite a few very important areas. I want you to start off with something which I think is going to be quite important for viewers to understand, because you went over it briefly, but it's worth looking at the philosophy side, not just the science side. So when it comes to Eric Baptiste, he mentioned that patchy taxonomical distribution was thrown out. And this obviously brings up the whole idea of theory ladenness and what role Darwinism plays in filtering the data. So could you just elaborate a little bit about this, please?
Yeah, I'm going to put that slide back up.
Sure.
So one of the happy but unsettling, if it's possible to put both of those adjectives in the same sentence, a happy but unsettling consequence of whole genome sequencing is that nature was able to speak for herself in a way that was unexpected, right? So when you get the complete genome of a microbial group, you often, and this is the case not just for microbial groups, but I'll stay there for just the moment because that's where Baptiste is making his main point on this slide. You have data that fit and data that don't, right? Or another way of thinking about this is some genes give you the tree you expect, other genes give you trees that you don't. What do you do in a situation like that? Well, Baptiste, as a phylogenetic, as an observer of phylogenetic method, notices that data are jettisoned to get over here. You start with this big buzzing mess of contradictory trees.
Sorry, Paul, you haven't shared your screen yet, so I think you're.
Oh, I am so sorry. I am sorry. And my cat is pestering me. I'm going to let my cat out. I'm sorry for the interruption.
No worries.
I didn't know he was in here. There he is.
It's a cute cat.
Okay. A thousand apologies.
No worries.
Put this up. Can you see that now?
Yep.
All right. I'll quickly recapitulate all the words that I said just a moment ago. You start with a messy original data set, some of which makes evolutionary sense to you, other parts of which do not. If what you want is a clean tree, maximally consistent among all the parts, that sort of fit your preconceptions. And I, as myself as a student of molecular phylogenetics, I see this all the time, that data are jettisoned, quite literally dumped out of an analysis along a path like this, where you drop out things that don't fit your preconceptions to give you the tree that makes sense. And this begins actually at the very first analytical step. When you have sequence data, they have to be aligned to build a phylogeny. So you're aligning genes for structural proteins, or you're aligning transcription factors if you're looking at an animal, let's say. And there are regions in the alignment that don't fit. And some investigators say just manually eliminate them, right? Drop them out, or figure out if that region is giving you a misleading signal, just eliminate it. This is highly problematic because it is taking nature and manipulating her. And really, we should be listening to her, if I can personify nature. And Baptiste says, as you'll see if you go to look at the complete talk, he said, this is a practice that really philosophically is deeply objectionable, because of let me give you, it's a silly analogy, but I'll give you a simple sort of story. It came to me a few years ago. Imagine I'm in the Vienna main rail station, and I'm waiting for my train, and it's not there, and it's not there. And I go to the station master and I say, my train to Budapest hasn't arrived. And he said, well, that's impossible. Austrian trains are the best in Europe. They're always on time, right? And I say, well, that may be so, but my train is, 2 hours late. And he says to me, oh, we only count the trains that run on time.
Yeah. That's a good example.
Yeah. So I think, in fact, what's happening in molecular phylogenetics, that is the use of molecular data to reconstruct the history of life, is many investigators are now waking up to the reality of these data manipulations. And they are troubled by them, and they should be. And I think that if I had to make a prediction, I think over the next decade, you're going to see an increasing number of papers saying, we can't do this. We have to take the data at face value and try to make sense of it without prejudging the outcome that we think must be so.
Yeah.
I think you will agree with me, Subor. It's the power of the theory of common descent in terms of its effect on perception, on biological perception, that because it's taken to be the furniture of reality, right? There was a last universal common ancestor. There was a tree. And the terror of creationism, so to speak, feeds into that, right? You're just going to get rid of data that don't fit.
Yeah, absolutely. And this is why it is important to highlight the philosophy side of things. So Darwin, in The Origin of Species, he also mentions, he doesn't call it theory ladenness, that was a label that was given later by philosophers, but he also mentions, well, if you don't start off with some form of a theory to filter the data, you might as well go to the quarry and count pebbles. So this is just a fact of life, that you have to have some form of a theory to filter the data. But once you don't use it to just filter the data, it becomes a permanent thing which is going to get rid of relevant data. Then that's obviously very problematic. Now, I wanted to ask you a counterfactual.
Yeah.
You said that, just near the end of the presentation, you said diversity may be functionally significant, and there's some very exciting stuff that you said is out there. What if diversity or those diverse elements are totally functionally insignificant? That wouldn't do anything to undermine the argument, right?
No, because they still have to be accounted for.
Yeah.
Right. One of the things about ribosomes that I think is so interesting is they represent variation at the biological limit, because there's no lower level.
Yeah.
If you think about variation of, like, my height or my eye color, or pick any higher level character in Homo sapiens, or my cat that I just put out, that variation is at a relatively high level in the organismal hierarchy, and it has multiple components that give rise to it, right? So many anatomical features in Homo sapiens have a polygenic basis. Multiple genes interact.
That's right.
To give rise to them. You go to the ribosome, there's no lower basement, right? There's no lower level. You are at the limit of variation, because below that is chemistry. So it's, in the case of ribosomal differences, they have to arise in a kind of quantum way. You've got to go in right into the machinery and change them. Which tells me that these differences, if they are real, may be the point at which the groups break apart, right? Because there's nowhere else to go. I'm sorry, I'm having a little bit of difficulty expressing the point.
No, it's actually very clear. It's very clear what you're saying. I mean, we were talking about the base element here.
The base element, because below that there is no more biology. It's chemistry. It's chemistry.
Exactly. And we should probably sometime in the future cover, because you said polygenic, the word itself triggered Dennis Noble's idea about how it's like an orchestra and how these genes interplay. Maybe that's something for the future. So there's something else I wanted to highlight, which is when you brought up Lars Vogt, in terms of there's elements which are of the theory which are untestable. So this goes back to the old sort of rebuttal that some Darwinists pose. Well, surely this theory is testable, because we haven't found rabbits in the Precambrian. But according to what you've elaborated upon in the beginning of the presentation, even if we found rabbits in the Precambrian, it would make no difference, because it's consistent with every possible, the universal common descent is consistent with every possible conceivable pattern that we can find.
Right. It turns out that rabbits in the Precambrian is actually related to the principle of continuity. And I think that that actually rabbits in the Precambrian are found all the time, meaning that fossil distribution is constant. The ranges of fossils change with some frequency.
Yeah.
You'll see a scientific paper. We found a chordate 50 million years earlier than we expected. Well, what's happening there? What's happening is that the evolutionary expectation is just goes out the window, right? And the anomalous position is accommodated by the theory.
Yeah.
However, it is possible to tell that story in a different way, where you would say, these fossil discoveries lead me to doubt that common descent is true.
Yeah.
What's happening in that case is you're taking your biological knowledge and you're saying the strain on common descent, the strain on this geometry is so great that it's breaking. And at this point, I have to let go. And there are examples actually of paleontologists who have done this. Gunter Beckley, for instance,
Yeah. Yeah.
one of my colleagues at Discovery Institute, has talks available on YouTube, but also things that he's written where he says the neo-Darwinian picture simply breaks down in light of fossil data.
Yeah.
So it is a funny thought experiment. What would happen if a rabbit were found in the Cambrian? What would be the outcome?
Yeah. I mean, it doesn't really matter because we've already had those rabbits in different forms.
Right.
Yeah.
Right. No, I'm just thinking specifically.
Oh, actually, if it was actually the case, yeah.
Yeah.
I mean, it's the way I see it is when Darwin wrote in The Origin, when Huxley asked him not to be so stringent when it comes to making gradualism an absolute assumption for the theory, and he basically said, well, the theory will absolutely break down. And obviously gradualism hasn't turned out to be correct in the fossil record. Well, if Darwinists can re-engineer the narrative after that, then the pre-Cambrian rabbits or anything else that they put out there would be cake, right? That would be pretty easy to do compared to how they've covered up the whole gradualism mess.
Yeah, I think that it's both a strength and a weakness of common descent that it is really a formal geometry, right? You can't falsify the form of a tree. It's a mathematical object, right? It's a Steiner tree with a single root. What you do is you presuppose a geometry and then you fit your data into that geometry. I think what's happening now, not so much with fossil data, but with molecular data, is biologists, as I discussed in the talk, are unwilling any longer to force data into a particular geometry. They're asking what might be the case in light of all the processes we know, such as lateral transfer and so forth, that could give us the patterns we see. And I've been at this a long time, and I'm very excited to see what will happen in the next 5 to 10 years as DNA sequencing accelerates, becomes increasingly inexpensive. I think I mentioned in previous talks that it's now possible, entirely feasible, for a high school biology class to have their own sequencers.
Yeah.
And to make some of these discoveries themselves. So it's really, biology is the science to be in, right?
Yeah.
It's golden age.
Absolutely. And I hope in your upcoming book you do highlight that, without, obviously no one likes praising themselves, so I'll do it for you, actually highlighting that you're the one who came out with this publicly in the early 2000s, saying actually these orphan genes, or what they refer to as taxonomically restricted genes, whatever you want to call it, they're just going to, increase as the years go on. And I don't know of anybody else who actually did that. And we don't find Darwinists even responding to the fact that this was a prediction which was confirmed.
Well, I think that if you go on the web and look for articles critiquing me from, let's say, 2006, 2007, I'm getting bashed about, and it's never pleasant to be attacked that way, but it's nice that that nature keeps her own counsel, and she's fiercely independent, and she's got lots of surprises for us. Which again, to me, I love what I do, because I wake up in the morning thinking, what surprise am I going to find in my collection of journals that I am looking at today, and it's great.
Yeah. So I have one last question. And this should be a brief one. We've gone over time. I'm aware there's quite a few people who they're going to be a little bit confused because they came onto the stream late. For some reason, more people jumped on maybe about half an hour into it. We can go back and rewatch it. So this is actually to do with something we previously discussed, that similarity is not enough to establish universal common descent. So Lauren Williams speaks about if you mess with the ribosome, it's basically death, and the three conditions that you laid out, that you need to have variation, you need to have selection, and then obviously a stable sort of hereditary processes going on. Well, when Darwinists are so keen to point out at anything that they, ignoring all the orphan genes and all the things that don't fit, but say endogenous retroviruses or similarities in ribosomes, couldn't we, rather than going over the problems with putting together the diagram according to what the data says itself, couldn't we simply say, based alone on the fact that variations to the ribosome would lead to non-functional ribosomes, your entire argument breaks down? So we wouldn't need to do the rest of, I guess, your presentation. Would that be sufficient to refute universal common ancestry?
I think it would pose a fundamental challenge, because as I'll be addressing in 2 weeks, on the topic on the 24th of similarity versus transformation, a statement of similarity is a static description, and lots of things can be similar, right? There's a famous passage in Shakespeare, in Hamlet, where Shakespeare is talking about the clouds, right? It's backed like a weasel or something. I don't remember the exact quote. We find all kinds of things similar to other things. Transformation, by contrast, is a dynamic pathway.
Yeah.
Typically, with a transformation that's well supported, you have some process known independently to be operating that enables you to move from natural kind A to natural kind B. Radioactive decay is a good example of this. But the alchemists were misled by the apparent similarity of base metal like lead to gold.
Yeah.
And they thought, well, it must be possible that there is a chemical pathway, a pathway accessible to chemical manipulation between the two. And we now know, don't waste your time, right? These are elements. Unless you have a particle accelerator, you are not going to be able to traverse that interval. The transformation that you hope to achieve is physically impossible. So if we could show or find a way of here's what I hope, and I'm actually hoping to talk about this next week. There does seem to be now a new technique for manipulating ribosomal sequences in vivo that would allow you to kind of explore the envelope of variability for that machine and say what variations are possible, meaning you have a viable ribosome with the following variations and which are forbidden.
Yeah.
That kind of knowledge is precious, because it allows you to separate possible from impossible, transformation from misleading similarity. Yeah. And this problem of giving these pathways reality instead of just saying, they look the same, they must be derived from a common ancestor by an unknown pathway of transformation. Excuse me. So I'll have more to say about that next week. But I think being able to say to an evolutionary biologist, that transformation is biologically impossible, don't waste your time, that would be very powerful knowledge if we could get our hands on it.
That's it. Absolutely. That's a good way to end the session. I'd just like to end upon something else I was thinking of while you were going through your presentation, which is, even with all these problems, we do have obviously some scientists who are challenging the standard narrative, but because of naturalism, they will eventually have to go back to trying to make it work. So the writing's on the wall, and we can't brush the elephant under the rug. The conversation will eventually have to lead to naturalism versus theism when it comes to the paradigm within which you work. Because if we stick to naturalism and all of this, if we extrapolate all these problems and they get worse over the next couple of years, it still wouldn't be a paradigm shift until we change the lenses. I mean, that's just something I was thinking of. What would you say to that?
Oh, I think you're right. I think that, and I addressed this, I've touched on it in previous presentations, that what you are able to consider as a possible cause affects how you perceive the living world.
Yep.
And naturalism, meaning, to unpack that term, meaning physics is the ultimate layer, and everything derives ultimately from physics. There is no such thing as mind or agency or creative intelligence. That puts a wall around your freedom as an investigator that I think nature itself or nature herself doesn't recognize. We impose that wall. And I'm heartened by the historical fact that the science of biology was born in a theistic cradle.
Yep.
In the work of people like Linnaeus and Cuvier and others. And if we can regain that metaphysics of explanation, I think biology, the science I know best, will flourish. We won't lose anything because we won't take anything away from nature herself. So you're right, Subor. Ultimately, this devolves to a question of what metaphysics of explanation are you going to accept? And my appeal to the listener, to the viewer, is choose the metaphysics of explanation that gives you the greatest intellectual freedom.
That's right. That's a perfect statement to for summarizing this particular talk. Thank you so much, Paul, for giving us your time. So everybody watching, please share this stream. This is now part 6. The references will be put upon the comments, and I'm going to pin that. If you have any questions, please send them to me so we can ask Dr. Paul next week. So until next time, Paul, thank you so much for your time. It's been a pleasure.
Yeah, same here.